Welcome to Protocol Particulars, the video interview series where we touch base with members of ASAP to learn the ins and outs of their protocols. For today's episode, I have with me Mujahid Azfar, who is a graduate student at KU Leuven and part of Team Vangheluwe. For today's episode, we are going to be talking about the protocol Fluorescently-labeled Polyamine Uptake via Flow Cytometry, which was originally authored by Marine Houdou, but a protocol that Muj actually has done quite extensively and so has volunteered to talk with us about. Hi, Muj, how are you?
Hello, I'm good. Excited
Good.
for the interview.
Awesome. So before we go into some of the details about the protocol, I thought we could just touch base about your team's ASAP project and how the protocol actually relates.
Right. So as you mentioned, I work for Team Vangheluwe, so that is headed by Peter Vangheluwe at KU Leuven, and it's essentially a project that brings together multiple groups from across the world, who all bring their diverse backgrounds and all their expertise together to understand a common problem that we have, and that problem is to basically understand polyamine and lipid homeostasis at a lysosomal level in the context of certain Parkinson's disease-related genes. And basically, it's in this context that we study how this homeostasis is achieved and how, when it goes wrong, that can contribute to disease-related phenotypes. And so this particular protocol in question is about measuring polyamine uptake using fluorescently labeled polyamines, and that helps us to basically understand how polyamine uptake happens in our various cell models.
So you mentioned that this protocol is using the fluorescently labeled polyamines and monitoring their uptake. Had there been other methods in the past that people used to monitor polyamine uptake in cells?
Yeah, there have been. The most commonly one that was used was using radio-labeled polyamines, and that's quite a robust way of measuring polyamines. It's very sensitive, more so than our fluorescently labeled polyamines, but it comes with its own challenges. Obviously, working with radioactive material, you can't, that comes with its own risks, and it also poses certain challenges of putting active cells through a flow cytometer or in a confocal microscope, and you can't, so using fluorescently labeled polyamines kind of circumvents that issue, and it makes it much easier.
All right, that's good to know. Um, so this protocol has four major components to it. There's like a prep before starting the actual protocol, some information on seeding the cells and incubating the cells with the fluorescently labeled polyamines, and then actually harvesting them for the flow cytometry. In the before starting section, something that was mentioned was that users should prepare an appropriate dilution of the fluorescently labeled polyamines for the cell culture medium. And so I was just wondering if you have any insight on like what an appropriate dilution would be, if you've seen that there's some sort of optimal range in your hands, or if you have any recommendations on that.
Right. So, well, that's a very relevant question because it's paramount that one studies the linearity of the uptake or the kinetics of the uptake beforehand to make sure that the signal you're measuring is pretty much in the linear range of the uptake. So to do that, you would do a dose response and basically test different doses and across your different cell models to see whether you see windows that you would like to see, whether it's between knockdown models compared to controls and stuff. So we typically use a 5 micromolar range, but that range, that dose we kind of chanced upon after doing extensive dose response studies. And that was really right bang in the middle of our linear range of our uptake, which meant that if we wanted to boost the uptake, we still had plenty of headroom to, increase it without reaching a saturation point. So different transporters may have different kinetics, different cell models. Cell lines may take polyamines up from the extracellular environment at different rates. So it will all vary from cell type to cell type, transporter to transporter. And that's why it's crucial to definitely test that before you use such tools.
Yeah, so it sounds like users will definitely need to optimize
Mhm.
that based on their conditions. So another question I had was that, you're working with fluorescently labeled polyamines, and so you note several times in the protocol, or Marine did anyway, to protect the samples from light. And so I was wondering if maybe you could just give your recommendations on how you go about doing that.
Yeah, well, it's just very simple. You just, I mean, our cell culture incubators aren't, when they're shut, they don't really have like a light bulb that's on, so it's like protected from light that way. When we are harvesting the cells after they've been incubated with the fluorescently labeled polyamines, we try to, keep the lights off in the room. And while we're trypsinizing the cells, we kind of, wrap our plates up in aluminum foil and things like that. So very basic things. That being said, I think it's more of a case of being better safe than sorry. Um, these fluorescently labeled polyamines aren't that prone to bleaching based on, I mean, they will at some point, but we'd rather not reach that point. So just basic common sense to keep it safe from light uh, when you can.
Yeah, that makes sense. And then, you mentioned incubation, so that kind of is leading into my next point. And I was wondering if they're in your hands, if you've seen like a minimum amount of time that people should incubate for. I think in the protocol it says that users would need to optimize. And so I didn't know if you'd seen a minimum or a maximum for being able to see a signal when you actually go to analyze by flow.
Yeah, that's again a great question, and it kind of ties back to the one of the questions we talked about earlier about enzyme kinetics. So we've got this dose range that you have to kind of search the optimal concentration within to make sure you're in a linear phase of an uptake. But also the time course can determine if you incubate them for too long, you may reach a saturation point at some point. So we also do a time course analysis. So we incubate these polyamine probes for different incubation times before we actually decide on what time incubation time is the optimum. So for certain cells, it's half an hour. For other cells, it's 2 hours. So it can vary quite drastically.
I think that's really good to know. And
Mhm.
then just thinking back to when you first started doing these types of experiments, were there any parts in the process that were particularly tricky that you could give advice on to others who are starting these experiments, or potentially if there's any like really critical steps that others should be aware of?
I think, yeah, I mean, definitely the dose dependency and the time dependency is an important one because if you read literature that uses such tools, quite often people use a single dose and one tends to mimic what they read in a paper, and then they might not see the phenotype they want, but that's probably because they're translating findings or a protocol from on a different cell line or a different transporter to what they are looking at. So that's definitely one. So definitely do that. Um, as far as it's quite a straightforward protocol. Certain cells, when you trypsinize them and harvest them, and then pass them through the flow cytometer, tend to clump together. Some cell lines don't. So I definitely experienced that issue in the past. And what I do that, how I kind of solve that issue is just by adding a little bit of EDTA to my resuspension buffer, which is essentially the buffer that the cells are in when they are passed through the flow cytometer. So it's around 2 millimolar of EDTA that I add. And in the protocol, I think we've written that it's a 1% BSA, bovine serum albumin, that we also add to this buffer just to keep the cells happy. But that can also sometimes make your buffer a bit viscous, which doesn't really help with clumpy cells. So you can also reduce that. Some of my cell lines do tolerate being with the BSA concentration being reduced to 0.1% even. So, but some cell lines may not like that. So that's also something you can kind of play around with. But apart from the clumping issue, which sometimes happens, but not that often, it's not that big of a issue, and it's not that trickier protocol.
That's good to know. So you covered quite a bit. But before we end, I just wanted to double check and see if there was anything else that you wanted to add?
No, not really. I think you were quite exhaustive in your questions list. So yeah, I think we've covered everything.
Perfect. Well, thank you so much, Moosh.
Yeah, my pleasure.